My name is Ahmed Mahfouz. I work at the Human Genetics Department at the Leiden University Medical Center. And I also have a guest affiliation at the Delft Bioinformatics Lab at TU Delft. My group mainly develops methods for single-cell data analysis. But yeah, so I'm here. I will only talk about the training that we do in terms of single-cell analysis courses. So just to give you a quick overview, in almost all the cities and the universities in the Netherlands, there are labs doing single-cell research at the moment, and I think of them as either doing computational developments, or they develop technologies, or they just apply these to answer questions related to their research interest. There are also multiple research facilities that offer single-cell analysis, and that also includes companies, by the way. We have been trying, I think, for the past 4 or 5 years to bring these people together. So we started with a meeting in 2017 just to see who's actually interested in doing this type of work in the Netherlands. We have been integrating single-cell sessions into the BIOSB. So BIOSB stands for it's the Dutch Bioinformatics and Systems Biology Research School, and this is our annual conference. There has been a lot of focus on single cell for the past 3 years or so with dedicated sessions. There are many groups that are part of the Human Cell Atlas Lifetime and also the Single Cell EQTL Consortium, which is based here in the Netherlands as well. And more recently, we started this network here in Leiden, where I'm based, where we have monthly meetings with people who are doing single-cell research in the hospital. So that's the LUMC at the university, LU, but also in the bioscience park that's attached. And there are plans already to make this sort of a national network, and that's not focused on education, but of course, education is a big part of these efforts. So in terms of training, there is one annual single-cell course, so I will tell you more about this, and that's mainly organized by Leiden and Erasmus and Medical Center in Rotterdam. There are multiple lectures that are given during different courses at the master's level, and that's almost everywhere in the different universities. I've also helped with the Elixir course that was organized by EA in 2019, and the course of MBIS earlier this year. And I do have to say these are very nice interactions as well, because it really helped us also set up our own course in a similar way. So about the big course that we run, it's a sort of a graduate course. So we aim at PhDs or postdocs who are doing single-cell research, but we also offer it to bioinformaticians that work in the facilities that I mentioned. It's an entry-level course, so there's no single-cell experience or analysis required before this course. But that also means we don't cover a lot of advanced topics. It is organized under the umbrella of MGC, so that's a graduate school for the South Holland, the part where we are, and BIOSB, as I mentioned, the Bioinformatics and Systems Biology Graduate School, and that's a nationwide graduate school. It's an annual course, so we have been running it now for the past 3 years. We do it in full, so we just did the course a couple of weeks ago. The first year was in person, the past 2 events were online, but that, yeah, of course, that was mentioned by many people here. The disadvantage is that, yeah, we don't have a lot of interactions. Advantage is that we can scale up quite a bit with the online setup. And I will not say much about this, but I have to say it is working quite nicely. We do miss the interaction with the attendees, but in general, I think it's working quite well. This is just a quick overview of the course. So we do a 5-day course, 4 days of teaching, and the last day we have a sort of a mini symposium research-like talks. We do mix lectures and lectures plus practical. So some topics we only cover by lectures. So at the beginning, these are covering different platforms, pre-processing and experimental design, multi-omics, and so on. But topics as QC, normalization, dimensionality reduction, clustering, and integration, there is a lecture and there is a practical afterwards. The first day and the first half of the second day are too much on focused on the protocols and how you generate the data. Later on, it becomes more and more on the analysis only. The practicals, these are sort of cookbook exercises that we give as notebooks. We do everything in R only, not in Python, which is something that, yeah, I also would like your input. We initially focus on R because this is offered to anyone who's doing single cell research. So life science researchers can also analyze their data, and I think R is a bit more accessible. But a lot of people are using Python at the moment. In our research, we are mainly using Python. So this becomes a little bit more difficult to maintain. In the first year, because we did it in person, we set everything up on the PCs in the room that we've been using. Since last year, we shifted to our Studio Cloud, and I have to say this is a very nice move for us because it makes it much easier to set up the course. We only have to set things up once, and then everything is ready. And if there are mistakes or things that or bugs that needs to be fixed, we can also fix that really easily. The exercise we now do in pairs, so we team people up, and that's mainly to address that point of the online course that people do not actually meet each other, and I think that helped a lot. All the materials are in this link, so you can check them out. And during the practicals, the TAs are available to answer questions on MS Teams or in the room when we did it in person. Maybe good to mention, so we don't have people dedicated for training, so this is all covered by us, the research groups, and the TAs are basically PhD students who are helping during that week. So a bit on future plans. So from our design, the course seems to be a little bit split between the wet lab and the dry lab part. I like it because I think people should know a bit how the data has been generated, but for some people, this is a bit difficult. So we are thinking to have a bit of a hybrid setup where you can sign up only for the first 2 days or the last 3 days, or the combination if you like. We're covering mainly only single cell RNA-seq, so but I do think that there is a big shift also in the field or the community to other data types. So single cell attack, for instance, is very common at the moment. So perhaps we do need to address this in the coming years. I would really like to do more advanced training if we have capacity. So I think it's nice to have a basic course or an entry course. But yeah, that does not, for instance, attract a lot of people from the computational community because it's not very interesting. You can easily find a lot of material online to cover these basic topics. And I'm really looking into these project-based courses where we don't have to do a lot of lecturing, but then people can work on their own problems. That's a bit my short summary. If you have any questions or input, I'll be happy to take.